Okay, good afternoon. I am Dr. Duangrudee Tanramluk. So I'm from the Institute of Molecular Bioscience, Mahidol University. You can also call me AjarnWi. So let's start with the basic bioinformatics lecture. So the objective of the class, at the end of this lecture, you should be able to describe principle and flow of bioinformatics data necessary for your research. And you should be able to identify bioinformatics tools used for sequence and structure analysis. And you should be able to describe the benefits of using bioinformatics for research. The content, first I will give an overview. Then I will talk a little bit about the central dogma and bioinformatics information. Then I will describe the types of bioinformatics data and characteristics. And then I will take you through the structural bioinformatics data and the details of the structures that used for bioinformatics analysis. And then I will give a bit of overview about bioinformatics databases and applications. So this is the video, The Inner Life of the Cell by Harvard University. Have you looked at the video of Inner Life of the Cells before in other class? Let's see. It is this one by BioVision. Is about when the cell was infected and what process occurred. So this is at the cell surface, and several transmembrane proteins are recruited upon infection. This is the cytoskeleton, microtubules. So you can see the scaffold of the protein. The structure will be created or degenerated upon the purpose of each protein production. So sometimes it was cleaved as it's no longer required that path. Sometimes it is built. So this is the microtubule that was built and degenerated. And this is the dynein or kinesin molecular motor that carry the vesicles from the rough endoplasmic reticulum to the Golgi apparatus. So when the protein is designed to be produced, it will be carried along the cargo. So this is the mRNA that was generated. And there is the ribosome to mRNA. And this is the protein that is being produced. This is when the ribosome attached. So the cargo of the protein production was carried along. And then some proteins are released, some are attached as transmembrane. In the white blood cells. So this is how we fight infections from various molecules. So the purpose of bioinformatics is we trying to link between the generation of the data from the DNA blueprint until it's transcribed into mRNA and translate into protein. And then we want to link between these three levels. And we want to make use of the data we gain. At the final step, we want to see what the protein are produced and how it's function and how the various networks are interconnected. So the central dogma, as already lectured in other classes, is the cycle of passing on genetics information when DNA is transcribed to RNA, and the triplet codon specify each of the 20 amino acids. And then after translation, the protein sequence will be synthesized. So you have the DNA and RNA and then the protein. So the reason when we talk about interlink between these levels is because we want to make use of the DNA data. And then, for example, if we got infected by the virus, we want to know how to combat it and how can we design drugs, or how can we understand the pathways or the molecular processes underlying the biological processes. So the types of bioinformatics data, normally for the DNA sequence, there are 4 base letters, A, T, C, G, with the length around 1,000 letters per genes. And this is when we talk about the DNA that encoded certain protein function, but for genomic analysis, there will be millions of data like that. But we want to know it at the molecular biology levels, where we can starting from cloning the DNA up to the protein. So normally we talk about something around 1,000 letters, and protein sequence data are strings of amino acids, 20 of them, and it's about around 300 amino acids or more. And structural data consists of more than thousands, maybe 10,000, or maybe even larger than that. And it has the X, Y, and Z Cartesian coordinates with it. So that's how the molecular visualization tool generate the pretty pictures that you see in the inner life of the cells. So we solve the structures from X-ray crystallography or from cryo-electron microscopy in order to see the structure of them. So the DNA sequence data looks like this. It will have the greater sign and some IDs, and then tell the species, and then tell the location on the chromosome. And then it will enter the new line and followed by the letter ATCG, something like that. For the genome browser, there's various genome browsers available, like this one from NCBI, the Genome Data Viewer. Raw DNA can be used to search for genes or for annotating introns and exons and for linking specific genes to disease. So when you open the genome browser, there will be several panels for you to zoom in and out at various levels. And when you go to the gene level, you can look at them in details, the letters inside, and you can analyze them. So normally the format that you use is the FASTA. And for the protein sequence data, they normally start with greater sign as well, and followed by some IDs. We will talk about these IDs later. And this is the Uniprot Code, stand for Universal Protein Knowledge Base. So it will start with the short name of the protein, such as this one, Cytochrome C _ species, like Cytochrome C _ human, and then some details, and then starting with the amino acid letters, 20 of them in the new lines. So this is what we call the FASTA format. They will consist of lines like this. So the homologous protein sequence are used for making multiple sequence alignment to find the functional size or for protein modeling. This is the example of multiple alignments. So the aim is to align similar amino acid in the same block, like this, in the same colors. Like like leucine. So they align the amino acids with the same properties on the same blocks. And if it's red or it's magenta, E is for glutamic acids. So they align E or D aspartic acid in the same blocks like this. So they will insert gaps in order to find the most suitable alignment. So they want to find the conservation and want to infer function from the sequence conservation. What we call the percent identity is used to measure how similarly are the particular amino acids sequence with one another. For example, this one, corn and sunflower, may be more similar than human, and human may be more similar to chimpanzees and rhesus monkey more than the corn and sunflower or the duck or chicken. And this multiple sequence alignment will be used to create the phylogenetic trees to find the relationships among them in the tree of life. So let's come to the structural bioinformatics data. So as you have learned from several other classes before, that the proteins start from the primary structures of A, D, E, something like that, from one letters amino acids into long string like this. And if these amino acids in the long string have repeating torsions and form secondary structures, they will have characteristics like in spiral shape called the alpha helices, or if it comes on the right and going back to the left like this is called beta pleated sheet. And if there is the combination of them, such as alpha and alpha or beta alpha beta, this will be called the motif. And if they comes into the recognizable shape, recognizable fold, we call it domains. They may be like prism domain or clams or sandwich or solenoids or whatever basket. They will call orthogonal bundles for square basket, for example. And then if they have functions, they will be called the tertiary structures. And if there is cooperativity among them, we will call it quaternary structures. So the building blocks consist of the amino acids in a long string that link together by the polypeptide chain like this. And this is the L amino acid, which is the left-handed organization. And when we look at these atoms that build up these amino acid side chains, we will look into the molecular viewer on the periodic elements table. So when you study general chemistry, the periodic tables look like this. And when you look at each atom in the molecular visualization tools, what you see is this. So we want to focus on this row. And this row, we want to see carbon in white, nitrogen in blue, oxygen in red, and sulfur in yellow. So carbon may be varied based on various proteins that you build and trying to create the suitable picture for your publication figures. But normally we use it white, sometimes maybe other colors, but the color that stay, normally nitrogen in blue, oxygen in red, and sulfur in yellow. So you memorize only 4 colors in order to make sense of the amino acid side chains. In this structural bioinformatics, we will look at it in the 3-dimensional orientation or conformation. So glycine, this one is the smallest one. It doesn't have side chains. It only have the main chain atoms. And it's the smallest and it's allow flexibility. For alanine, leucine, isoleucine, and valine. This one is hydrophobic amino acids. It's only serves structural role. It has uninteresting characteristics So it's only serve structural purpose. For serine and threonine, these have OH. So this hydroxyl oxygen serve for post-translational modification, such as in protein kinase. They may add phosphate group here. So this is allowed for other reaction to occur. For cysteine, this is reducing agent. As you know, it can form cysteine bonds. So you can see that the yellow color is cysteine. The yellow only have in cysteine and methionine, for example. And this one, it has two red tips. So it have OOH. The position may be swapped, but from the X-ray crystallography, we only see the oxygen atom without hydrogen. So we don't know which flip side it is, depends on the nearby residues. But if it have two red tips, we will know that it have partial negative charge characters. So it normally have negative charge, and this negative charge from salt bridge with the positively charged amino acids. And this one, polar uncharged, you can see the one blue, one red. So it makes it polar, but it doesn't have charge. So asparagine and aspartate have exactly the same size, but the atomic type are different. And glutamate and glutamine have the same shapes. I mean, static, but when it's in the three-dimensional structure, it may rotate around with several rotamer. But normally in 2D drawing, it draw pretty much have the same size. But these two atoms are different. So glutamate, glutamine, aspartate, asparagine, the shape is similar, but the atom types is different. So for methionine, this one is used for when we solve X-ray structures, such as in selenome methionine side chains. We want to use it to make help while solving X-ray crystallographic structures. For lysine, arginine, and histidines, this one you can see the blue colors. The blue colors mean it have partial positive charge characteristics. So the blue colors form salt bridge with the red colors. So these ones will be used for like epigenetics, or sometimes in tryptic digestion, it will recognize the positive charge amino acids. And for proline, it will form king in the helix as it rotates back. So this is the main chain nitrogen. And for phenylalanine, tyrosine, and tryptophan, these ones are aromatic amino acids or large hydrophobic amino acids. It will be used for molecular recognition, such as for chymotrypsin, it will recognize large hydrophobic amino acids. So these ones sometimes is called having pi-pi stacking interactions, as it have pi electron and it stack like tire, like car tire, stack on top of one another, or sometimes in the shape like this. One is on the horizontal orientation, the other one is vertical on top of it. So these one are for pi-pi stacking. In the assignment, I will ask you to recognize the amino acids from the shape and tell me the characteristics, what it does. So for the protein size, in trypsin inhibitor, it may be as small as like 58 residues, or 36 angstrom apart. This is botulinum toxin. This is Botox that we use to inject into our skin to make the face look nicer. So this is how it looks like. And the size may be as small as trypsin inhibitor or medium size like botulinum toxin, or maybe extremely large, even larger than 4,000 residues or in mecadaltons. And sometimes the nucleopore complex is even larger. So with the advent of cryo-EM, the structure size can be unlimited. The molecular machinery. So this is the rough sketch of how the protein have various size and shape. So as you see in the video, the protein can adopt various function. So it's help with how the structure look like, how the structure allows it to do that certain function. For example, this one in GROW-EL, this is the chaperone protein. It will have a tub-like characteristics, look like barrel, and then you can put the nascent protein inside and close the lid and shake and then make the protein fold correctly, for example. So it's have the tub size shape in order to allow other protein to get inside and put the lid on top. And this is like virus and electron transport chain that I will talk only a bit about Cytochrome C, as you have learned earlier in Ajahn Chanan's class already. This is ATP synthase. And these are the other protein on the electron transport chain. And what else? This one, as earlier we talked about kinesin and dynein that walk along the microtubule with little feet. So it has this little feet shape, and then it carry along the cargo, the big cargo. And this is the microtubules. So it comes in various sizes and shapes, pretty unlimited. And now there is the project called AlphaFold that for 200 million of the different shapes of proteins available. So we are in very exciting era to see all the structures solved with the help of AI and DeepMind. So Google built a project called DeepMind, and then they solved the protein by helping the protein folding problems. They generate the server AlphaFold to solve the 200 million protein structures. So the Pdb file format, in Pdb file format, when we want to open the pretty pictures, it will start with the header line like this, and they would tell what the content of the protein inside. So this one, they will say, okay, this protein that you open the file in one stc.pdb have cyclic AMP dependent protein kinase containing in this file. And then the header will also say what are the compounds. So they say this is cyclic AMP dependent protein kinase molecule, catalytic subunits, and have enzyme commission number, and then tell whether the protein is engineered. And then this is the fragments of the peptide inhibitor inside, and tell you the source of the protein, like this one from Bos Taurus come from cow, and tell the organs, and then tell you the expression system. So it was expressed in E. coli. And then after you finish the author line and details, so this one is created by Eng and Huber. This is the author's name who created this protein structures. And then after finishing the author line, it will start with the atom line. The atom records will have record of atom in the header, in the beginning of the columns. And then it will have sequence number, the atomic IDs in serial number. And then it will show you each atom. So nitrogen, carbon alpha, carbon, something like that, and then glutamic acid. So what this mean by glutamic acid? So it will have the glutamate pictures that I showed earlier in the amino acid side chains. And then these amino acid residue have residue number 86. And this X, Y, Z coordinate is the location of each atom in 3 dimensions. So the molecular visualization tools that I will probably teach you in the class protein visualization, it will generate the pretty pictures for these atoms. So the flat file look like this, and it just tells you the location of atoms in X, Y, Z coordinates in Cartesian. And then it will draw the picture that you see earlier in the video. For the sidechain representation, so this is the line representation. You can see that if it is the alpha helix and has this one glutamic acid 86, so this one glutamic 86. So this is drawn as this one glutamic 86. Glutamic 86. And if you draw it in line representation, it look like this with the colors. So it's a little bit better than the rough sketch, but it used to communicate about the functional group easily among chemists. But for crystallographers or for drug researchers, they will normally use the stick representation or the ball and stick representation. So if the radius of the ball is equal to the radius of the cylinder, it will be called the stick representation. And if they are presented truly as the sphere with van der Waals radii, they will be called CPK spacefill. So that is 4 representation that I introduced: line representation, stick representation, ball and stick, and CPK spacefill. So in the sidechain representation, sometimes we omit or not drawing certain residues, but showing only the secondary structures, like showing only the alpha helices or showing the beta sheets, and we discard all the side chains. And you will have pretty picture like this. In C alpha trace, it's easily that you can see it in various units, like this one. If you show 1 color per chain, you can see 6 units by just glancing. So this is the purpose of C alpha trace. It's easy to draw. So when I study my master degree, one of my exam is to draw hemoglobin. So it's pretty amazing how people in that generation can draw hemoglobin on the paper. So there is certain fold, and there's people that can actually recognize the fold by just using pen and pencil and drawing. But with this era, we can draw the exact shape using just molecular visualization tools. So this cartoon representation, we will just draw everything as ribbon and then use different colors. So this one in cyan is beta sheets, and the red color is alpha helix. And for schematic, it's pretty similar to cartoon, but it have the direction. So it will have the rocket shape in red color like this. And then the rocket will have the shadow like that, and then the rocket shape will be the alpha helices, and then there will be the beta sheets that look like this. So in schematic, it will have direction. For van der Waals surface, the purpose is to show the boundary of the active site, like when we use for drug designing. When we design drugs, we want to see the exact boundary and the location and the size and the shape. And then we can also add up the dimensions such as the charge characters, like here, the positive charge in blue, negative charge in red. Like that. So this is the detail of the C alpha traces. You can see that in C alpha trace, we draw the alpha helices without the side chains. So all these, we just didn't draw it. We just draw the alpha helices, and it will call the C alpha traces. And for solid ribbon, if you zoom in, you can see that the way they draw it is by thickening the lines of the peptide bond and make it thicker. And then they omit the side chain as well, and it will become the structure on the left-hand side. So if it's all the ribbon, like ribbon for helix and ribbon for side chains, and then it have green colors for turn. So this one is called the cartoon or solid ribbon. For schematic, you can see that the key is direction. So it will go from N terminus to C terminus, from N terminus to C terminus. For van der Waals surface, is normally have the radii according to the van der Waals radius. And sometimes you can use carbon in gray or in white, and nitrogen in blue, oxygen in red. Sometimes you can also color by pKa. So normally you use positive charge in blue, negative charge in red. And then if you are the drug researcher, you would try to generate the ligand atoms that fit in shape, size, and charge characters. So the shape have to be fit in three-dimensional structures, and the size should be suitable. So for drug virtual screening, just the size alone, you should screen like a half of them at least away, and then you have certain heat with suitable size, and then you can screen for the shape and then screen for the charge. So in drug design, normally if this protein have functions such as if it's allow for cell growth, and if you can design the inhibitor that blocks the function of this protein, and then it cannot make cell grow. So that's how you create cancer drug, for example. But the process is very difficult. But then let's put that to the next class that I teach about drug design, and then we can go into a little bit more detail about drug design based on the structures. So is there any questions so far? No? Okay, let's continue. So for rational drug design, you start from having the structures from X-ray crystallography or NMR. So you can see the overall shape by cryo-electron microscopy. But in order to see the location, the exact location of the atomic location, you need X-ray that tell you the precise location of atoms. And then you will pass the structure on to the computational chemist in order to do drug design analysis. And then The computational chemist will pass on the sketch to organic chemists to synthesize the inhibitor candidates. And then it will undergo in vitro testing, which is testing in the test tube, in vivo testing, which is testing in cells, and pharmaceutical chemistry testing, which is testing in animals, such as from mouse, rabbits, monkey, until the human population in clinical trials. So the process repeats so many times, and the process may take up around 17 years for one drug and cost around 1.3 billion US dollar per one drug to be discovered. And structural bioinformatics is here. So we start from learning target enzyme structures and help with this process of drug design analysis to help computational chemists understand better the structures of the protein. So the active sites and clef, this is the example of the protein kinase active site. Normally this site, the pocket is for ATP, but it allows for so many other drugs to sit inside. And since the protein kinase is allows for cell growth, cell differentiation, so by blocking the mouth of protein kinase or catalytic cleft, you can control how the cell grow, and then you can design cancer drugs, for example. So these are experimental drugs in comparison with the ATP structures. So Glycine is a wonder drug for treating chronic myeloid leukemia. In a nutshell, we want to find the blocks of similar sequences based on the following ideas. So we want to find similar sequences of amino acids based on these ideas. Phenotypes is the results of environment influence combined with genotype. And the genotype is inherited together with any mutations and then pass on to subsequent generations. So when it pass on the genotype, the mutation sometimes is being retained. By this way, if it's from the same lineage or from the same clades, it will show that certain species have the same mutation, for example. So through evolution, basically we want to find that which DNA mutation cause aberrant function which results into the diseases. And we also make assumption that a pair of similar genes can give rise to similar protein function if the genes are diverse from common ancestors. So what I mean is this, we want to find the sequence and then also several other sequences which are similar, because these similar sequences show that they should adopt the similar function. So the protein with similar sequence can be divided into two groups. One is called orthologs. So if homologous proteins are diverged from one another after speciation into different organisms, we call it orthologs. If the sequences are related as a result of gene duplication, it's called paralog. So orthologs such as Cytochrome C in human, Cytochrome C in birds, Cytochrome C in turtle, they are after speciation, after becoming the species. So orthologs is like one protein in different many organisms. And then if each organisms all have the same sequence or very similar sequence, they should have the same function. And for paralogs, it will be the protein inside the same species, but with different variants. So the paralogs also have related function, but it is a diversification of the same protein inside the same species. So it's like the same protein, same enzyme, but in variation of it inside the same species, such as in human, we have certain protein, and then we have hemoglobin beta, something like that. So when amino acid sequence similarity is high, we infer that it adopts the same protein fold with the known genes. So basically, we want to find the amino acids that are very high in sequence similarity to infer that they have the same function either in the ortholog fashion or in the paralog fashion. So the information that can be derived from protein structures, if you know the protein structures, the information that you can gain is like the crystal packing that show you the interface of the protein. The fold will tell you the evolutionary relationships, and the composition of the surface can help you design antigenic sites for surface patches, like for antibody. And the shape and electrostatic properties allows you to understand the cleft or active sites in order to design drugs. And the location of mutants and SNPs and conserved residues can also be shown using the protein structure. So you know which location the mutant is occur, whether they are buried inside the core of the protein or they are at the surface of the protein. So if the protein seems to interact with other protein, the mutation at the two protein interface will allow you to infer whether the function is different, whether they can be a point of study for certain disease because that mutation affects how the protein interact with the next protein in the pathways, for example. And protein ligand complexes. Ligand is basically the chemical compounds or the cofactors. So it's basically chemical compounds that are small enough to be sitting inside or around the protein, but it's not protein and not nucleic acid. It can be drugs, metal groups, or it can be vitamins, or it can be other chemical compounds, ATP, GTP. These are all the ligands. A look of the analogy of DNA sequencing video. So you may have learned it other ways around, but let's see this first, whether you understand.
The first method of sequencing the genetic code was devised by Fred Sanger. To sequence the DNA, it must first be separated into two strands. The strand to be sequenced is copied using chemically altered bases. These altered bases cause the copying process to stop each time one particular letter is incorporated into the growing DNA chain. This process is carried out for all four bases, and then the fragments are put together like a jigsaw to reveal the sequence of the original piece of DNA.
So this is how we obtain the DNA sequence letter. And in genomics analysis, we basically want to observe certain letters which are aberrant in certain population, such as the single nucleotide polymorphisms that causes the disease in certain group of people who got the disease. And the very basic thing of bioinformatics is if you are given a certain protein or DNA sequence, how can you obtain the piece of the DNA or the piece of proteins that matches with the input sequence? So the very basic thing you can do is called BLAST. So you can either do nucleotide BLAST, which you input nucleotides and getting back nucleotides, or you can do T+N, where you put the protein sequence and get the translated nucleotide sequence back. Or you can do the protein BLAST, like you put in the protein and get another protein in other species back. For example, if you have protein kinase, you want to blast it and see several other kinases with similar protein sequences. And you infer that if the protein have similar sequence, they should have similar functions. So I will demo how to do that. But in order to be a successful bioinformatician, you cannot just sit in the lectures. You have to go out and do things. So you have to do the hands-on practice in order to be a successful bioinformatician. So it's pretty much the practicing and skills that you learn after several training in order to do that by yourself. But I will give a dry lab by showing how I did it first. So for BLAST, this is how you do it. So for T BLAST N, you put in the sequence, and then you put the FASTA format, FASTA that I tell you earlier with the greater sign and then the sequence. So you blast it, just push the button, and then you wait, and this is how you get the output. So it will show you that this protein is cytochrome C, and then shows you various other cytochrome C with E value number. And then if you click into the source, you can see the DNA of that output sequence. So this is example how to blast. So by that way, you can put in your sequence and get out other protein in other species. And another resources that I very much want to introduce you is called the Uniprot KB. Uniprot come from Universal Protein Resources. So Universal Protein Knowledge Base. This is how you get the information of certain proteins, such as if the input in this class, I will use cytochrome C a lot because it's a small protein, and then I will show in details later on. So the cytochrome C _ human can be the query for your Uniprot. You can put in cytochrome C human, or you can put the IDs, or you can put these accession numbers, and then this is what you get after searching through the Uniprot. So it shows you the Uniprot ID cytochrome C _ human species, and show the functions of the protein, and then shows you the binding site, the position of amino acids that bind to heme and iron, and gene ontology. Gene ontology, I will tell you about gene ontology in the next slides. And then it shows subcellular location, where it comes from. So this one is from mitochondria. And this is the drug bank, which drug bind to this cytochrome C. And then shows you the various IDs. So in bioinformatics, everything is about ID that link from one database to another databases. Normally it comprises so many IDs, but you just need to know which IDs that you want to get from which database. And this is how you get the FASTA sequence. You can go to Uniprot and download the FASTA sequence. And it shows you the sequence databases. So how can you make sense of all these things? So let's remember it's about the IDs. If you get one ID, such as if you get Uniprot, you can hop to many other IDs from this linkage by this one page. So you can click on these items based on the catalogs of certain bioinformatics resources. And in order to perform the research, you need to know which database that you are clicking and the terminology that you use. So Uniprot can also allow you to blast other cytochrome C in other species, such as you can put cytochrome C _ human, and you click BLAST. And then it will show you all the cytochrome C in all the species in the world. So this is the output. It shows expected value. So the smallest is the better because is the parameters that used to measure what happens by chance. And if it's not by chance, the number is high. If it's e to the power of minus very large number, it means the value is very small. So this is cytochrome C in rat. In mouse, for example, we want to look for e value very small. And then from getting that sequence, you can also make the multiple alignment by clicking on this align button. But in real life, what bioinformaticians do is normally by command line. So normally they have a Unix workstation or Linux workstation in several various flavor, such as SUSE Linux or Ubuntu Linux or for Mac machine, and then they code it into the line codes. Doing BLAST, doing alignment, doing anything, they can do it with the command line. So this is how you do it by interface. It's easy for beginner, but if you want to take a serious career in bioinformatics, you need to learn the Unix command line and need to know a little bit of programming in order to get started. So this is just by rough ideas first, how to do it. And this is the primary database of the structures. So the primary database is called RCSB Protein Data Bank. So this one is in America. There is also the Protein Data Bank in Europe, which the PDBE or Protein Data Bank in Europe syncing to have exactly the same data as RCSB. And to the Protein Data Bank, there would be the 4-letter codes that specify each protein structures. And there is also PDB sum, which is the summary of the protein data bank data. So you can input the 4-character PDB codes, or you can search by the sequence by putting the sequence here, and you can get the structures back. And it will show the details that I tell you earlier about the PDB file format. So it has the title, the source from cow, the structures, the expression system in E. coli. So this is the digestion of the header of the PDB files into the pretty pictures. So you can see it from PDB sum, and it shows you the organization of the secondary structures on the sequence, and shows you also the ligand binding site as well. So this is how you get into the protein data bank file quickly to see what's inside. And there is also the derived database. So RCSB or PDBE is the primary data source. So exactly as the same as the crystallographer deposit it. But for the derived database, they process it further in order to suitable for certain purpose. For this one, CAT database stands for class, architecture, topology, homologous superfamily. So they process the structure and divide it into class, and within each class with what architecture it looks like, and its topology and its subfamily. So the protein structure, they divide it into 4 main classes, have alpha helix, most of them have alpha helices, or most of them have mainly beta sheets, or mixing of alpha and beta sheets, and have few secondary structures. So when you get inside the CAF database, they analyze your protein and say, okay, this one have mixing of alpha beta sheets, alpha helix and beta sheets, and inside the class have row yam roll, sandwich, prism, box, propeller. And so it subdivides from class 3 to class 3.10 as roll, or class 3.80 alpha beta horseshoes. And then 3.80.10 under horseshoe, there is also the leucine-rich repeat homologous superfamily, for example. So it's go to 4 different character: class, architecture, topology, homologous superfamily. So each protein, they digest it into the shape that are recognizable by each class. So when you search the protein data bank, you don't need to search by its function or its name or its species, but instead this one, you can find what protein have the propeller shape, for example, what protein have the prism shape, and then you can do my assignment on making physical model of the protein. You can fold the protein based on certain shape, for example. Ontology is the control vocabulary that is limited, explicit, and carefully defined set of terms. It's used for distributing queries among several databases. And by using ontology ID, you can batch query in batch with specified function and biological meaning of the proteins across various databases. So as I told you, bioinformatics consists of so many IDs. One of the IDs that important for you to search batch query things is the gene ontology. So the ID will have its special formats. In this case, gene ontology have GO colon 0004672, GO colon 7 numbers. So what it does is gene ontology divide into the 3 domains. One is the biological process. One is the molecular function, and one is the cellular location. So by dividing into 3 classes, you can search certain things based on gene ontology, which have is a relationship from one another. So basically, seeing this scheme, it go like this. Protein kinase is a go up, phosphotransferase. Phosphotransferase is a transferase. Transferase have catalytic activity. Catalytic activity is a molecular function. So everything dissects down to the bottommost, which is the subset of the top levels. So you have molecular function, biological process, and subcellular location. So it's go from bottom up to the top. And each level have its own GO, which is the dictionary that you hop along the database based on certain GO numbers. When you do this, for example, if you want to search the structure of protein kinase activity, you would go by GO 0004672 to find all the protein in the world which have the protein kinase activity using this ID. So you can go to ASSB protein data bank and find GO 0004672. And then you don't need to Google one by one. You can have the category of the whole protein in the universe, which have this protein kinase activity using just this ID. And so this one, you can see the layer. You can go from bottom up to the top. Is from protein kinase activity is a phosphotransferase. Phosphotransferase is a kinase activity. Kinase activity is a transferase, up until molecular function. So the top function have 3 domains, and each one have specified GOIDs. Next, forget everything. Now we want to go into network. Network of everything, we have a database called KEGG, Kyoto Encyclopedia. So it's the encyclopedia of biological pathways. So you know that protein doesn't work by themselves. They're interrelated with one another. And KEGG biological pathway link you from protein in the same pathways together, and they have maps like this for certain KEGG ID. And they curate the data basically for every protein, and then they put into the map like this one. I will show you the server that I built to link all these databases together. You will see how it does. So in KEGG, you can also see some pictures, but normally it just comes into pathways like this, and you can highlight the nodes like this on your pathways. Let's come to bioinformatics applications. So the case studies is Cytochrome C. I talk about Cytochrome C everywhere. So if you want to do research about a particular protein, let's come to the example of Cytochrome C, how we do it. So in Prentice Hall textbook, it shows you electron transport chain like this, and Ajahn Chanan may show you a little bit more about other videos. So Cytochrome C is basically what hops from this to this cytochrome BC1 complex to cytochrome C oxidase. So that's one is drawing. This one is 2Ds, maybe 3, but it cannot rotate. So for 3Ds, let's see here.
Protein-rich matrix contains enzymes necessary for cellular respiration, a process during which carbon fuel molecules are oxidized and reduced electron carriers are produced. Invaginations of the inner membrane, called cristae, contain 4 large protein complexes that harvest electrons from these carriers. Complex 2 accepts pairs of electrons from succinate and transfers them through a series of redox centers to coenzyme Q. The lipid-soluble coenzyme Q is reduced by electrons harvested from complexes 1 and 2, and then diffuses through the inner membrane, transferring its electrons to complex 3. In complex 3, electrons are transferred through 2 distinct series of redox centers, which allow them to cross the inner membrane one at a time. Finally, electrons are accepted by cytochrome C, which carries them to complex 4, where they are transferred through another series of redox centers to their final acceptor, a molecule of oxygen that combines with hydrogen ions to form water. Electron transport in complexes 1, 3, and 4 is coupled with the pumping of protons from the matrix to the intermembrane space. The resulting electrochemical gradient across the inner membrane is called the proton motive force.
So
Protons flow back into protein-rich matrixes intermembrane space, come to 4, where they are transferred through another,
now
which allows
we will
cytochrome C,
look at
which
this
carries them to
cytochrome
complex
C.
4, where they...
So cytochrome C is a small protein. It consists of 104 residues. It is a monodomain protein, mainly comprises of alpha helices and reverse turn. And it has a basket shape. So it's square basket we call orthogonal bundles. So when we superpose 4 structures, yeast, cytochrome C from rice, cytochrome C from tuna, cytochrome C from horse, you can see that it's superimposed or overlay almost perfectly. So it's very well conserved both in shape and in sequence and in the 3-dimensional folds. So you can see inside, this one is the heme, and this is how it accepts electrons. So cytochrome C acts as electron shuttle to hop from cytochrome C BC1 complex to cytochrome C oxidase. And when we want to use cytochrome C as query, we blast it. And after we blast it, we can do multiple alignment like this. So you load the sequence, and then from blast, you can save the series of cytochrome C, and it comes scrambled like this. So the way to do it, if you want to know which blocks is conserved or which block equivalent to one another, you will just do the alignment. So you want to insert gaps into certain parts. So after you insert gaps, you can see the cytochrome C multiple sequence alignment like this. And then you can infer which one is equivalent to which one. So in this one, you can see certain cytochrome C, like in human, have 105 up to this position. This one to 104. So you see if it doesn't have gap, only this one is one residue less than the top one. This one is 105, something like that. So each lines, you need to restart the number with the particular labels according to its own sequence. So this one go up to 105, and in the next page, it go from 106 and so on. And that's how you count which residues numbers they are. And through evolution, cytochrome C is developed early in the evolution of life. It's truly homologous, and it was shown that the variation is related to the phylogenetic relationship in the tree of life. It's found in all the 5 kingdoms of all aerobic organisms. So if you do the multiple alignment, there is a button that you can click to generate phylogenetic tree. So by that multiple alignment, there is one magic button that you click and you get this tree. And how can you learn what is this tree? Normally, we cut the tree like this. So when you cut the tree, you can see that this one is a clades of birds, duck, chicken, penguin. So when you cut, you can see this one have birds. This one is fish. This one is a mammal, human, chimpanzee, rhesus monkey, and it can go down to like corn and sunflower later on. So normally you cut the tree and then you classify them into family of related species. And that is a more complex tree, like circular tree or some bushy trees. So in cytochrome C interaction sites is mainly hydrophobic patch. So this one mainly hydrophobic, but it has the cytochrome C, it has positive charge residues to interact with the negative charge surface of this cytochrome BC1 complex. So in order to hop from cytochrome BC1 complex and cytochrome C oxidase, it hop and then it pick up electrons. So this cytochrome C molecule have blue colors, normally have positive charge. So it catch with the negative charge on the bottom, and then it transfer electrons. So that's how it go, and then it recognize certain place by having the charge. So in the old days, when people want to construct the protein structures, they use homology modeling. So they thought that protein with similar sequence often have common 3D structures. And normally it goes from having identified the homologs and superpose them, and then finding these known structures and build the atomic coordinates, cores and loops and side chains until you get the whole structures. But now the situation is different. We have the AlphaFold that I told you earlier. the protein folding problem has just been solved this year. And just last month, 200 million protein compounds was the 200 million protein structure, not compound, just structures, was deposited in the EBI website, EBI, European Bioinformatics Institute, EMBL. So they find the structure of the protein using DeepMind and artificial intelligence. And then we will see a very exciting era of having these structures available. Because you know in the previous chart that once we know the structures, there are many things that we can learn from having the structures. Case study 2. I will show you a little bit on spike protein. So let's forget about Cytochrome C. Now we go to spike protein of coronavirus. So when we talk about virus, this is what structural biology think of. It's like globular shape with several other structures inside. Many shapes, many sizes, many characters. Let's look at HIV for simplification of virus first, and then we go to coronavirus later. It shows the organization of the virus. So inside the HIV, this is the size of HIV in comparison with the T cells. And when the virus infects to the target cells, it will roll around the surface and its receptor catch with the target cells. And then it will recruit several associated protein and use its alpha helix to punch a hole. And you can see the process. So this video is from Janet Iwasa at Science of HIV. So it's punch a hole as red helix inside, and then the membrane is fused. And after fusion, it will put its genetic materials inside the infected cells. And the way to combat HIV may be if it really requires this blue molecule, we may be able to generate small protein like this green molecule we call peptidic ligand, right? And then when it wants to fuse the two membranes together and inject its genetic materials inside, the process can be prevented because that green peptidic ligand doesn't allow for the fusion of the two membrane like previously in the blue molecule. So this is one type of drug like protein subunit drugs, right? And for spike protein, this is another different types of protein. It's from coronavirus. So let's look at the video I built last April. Dr. Duangrudee Tanramluk at Methionine University. I'm reporting my mapping of the West Bengal B1618 mutations on the spike protein structure of coronavirus in the environment. So the past 60 days, you can see that B1618 occurrences here. And B1618 have 3 important mutations, which are deletion at 145, 146, E484K, and D614G. So what they mean? Coronavirus got the word corona from crown. Crown is this red part, which is called the spike protein. This spike protein can be understood from the structural perspective. You can see the red part here, which is the spike protein. If we flip it up, we can see the receptor binding domain membrane. And the South African UK variant has mutations in yellow, and the California variant have mutation in cyan. if the true combine at E484Q plus L452R, it becomes the B1617. And if the combination of E484K plus D614G in pink and deletion at the two green location, it will become the triple mutation West Bengal variant. So you can see that the structure is very beautiful and the beauty is extraordinary. However, it may mean hidden, scary consequences. So you can admire the beauty a little more time. The consequence of spike protein in B161H lineage comprises the deletion and E484K, which are found to have immune SK variant, and D614G, which increases infectivity. That is the example. And let's see what happened. If you look into the sequence from outbreak.info, you can see that it has ORF and it has the spike section, the spike protein. And then in the map, they will tell you the mutation location from which amino acids to which amino acids. So E484K, like here, normally is E glutamic acid mutated to K lysine. So you know what letter amino acids, right? So it's mutate from glutamate to lysine, for example, and then it changed the infectivity. So in this map, if you want to look into the molecular visualization, you can map it to the variant of concerns, mutation of concern like this. So B1617.2, this is the B delta variant, the delta variant. And in this case, I study the India variant, so it's the West Bengal variant triple mutation B1618. It said at position E484, it changed from E to K. And at position 614, it changed from D aspartate to glycine, for example. So we open the file 6ZP2 in the molecular visualization tools, and then we can map the location. So this one will be taught in the molecular visualization class. So basically you open the file with the Pdb format, and then you sift through the sequence line and find the position residue number E484, and then you highlight it in colors. So the reason why this is important is like this one, the Weiser lab by Longxin Kao and Brian Coventry, they designed a mini protein that binds to the receptor binding domain here. And then it prevents the virus from enter the cells. Or they can design the tripod like this on top that link with the 3 RBD, 3 receptor binding domains, and then it stops the virus in multiple variants altogether. So it's a very efficient way to combat multiple variants at the same time by having this kind of structures and then recognize the part that shares among several protein variants. So this is the spike protein, can you see? So so my expertise is in protein kinase. I basically study the protein kinase to find the dimension which should be contract or which should be expanded in order to design a suitable drugs for protein kinase. And I also built the platform in order to help drug design. So this is the platform. It links from chemical biology to structural biology to system biology. by having this, my platform can link from ligand to protein and to cake pathways. And then it have big data backend that link to the Uniprot and sequence databases and open target databases, and also help with the drug design research. For example, in here, you can put Uniprot code like protein kinase C, LPCT_human, like this, and search. And then you can search into the ligand structures. And then you can also search from the drug molecule, such as you search for staurosporine, like this. You can search anything like methamphetamine. Look up. See, it will help you convert the structures. Let's have a look at my test case. And then go here. So by having the query like this, you can see that the drug can bind certain protein. In this case, we go for the drug called staurosporine, which is a kinase inhibitor. When you look into the Manora server, you can see the Uniprot code, which protein binds to this drug. And this is Uniprot code and KPCT_human. This is Uniprot code. This is pathways. You can see that from autophagy pathways like this. I allows you to link the ligand to the pathways and highlight which node in the pathway it is. So this one is how you go to search the pathways that this drug involved with the protein. And you can see the protein by clicking on here. You can see the Uniprot, or you can click on open targets, which will lead you to the page that shows you which organ it affects. For example, you can go to baseline expression. You can see that this drug go to the protein, and this protein expressed in blood and in immune organs. And you can see the structures. Like here, you can link to the protein data bank. Just click inside here, you can visit the protein data bank. And it also shows you how tightly the drug would bind. And then it shows structural conservation. So this one, it shows the superposed structure of all the protein that binds to the drug called staurosporine and show the location. So the blue part is the very conserved parts. The green part is moderate and the yellow part is flexible. And then you can also see deeply into the details, such as you can see the hydrogen bond that affects how tightly the drug would bind. For example, you can click to this, and then you can see the hydrogen bond that interact with the drug from the protein 1XJD. So this is PDB code, and it tells you how tightly the drug would bind. So if you sort from tightest binding to the loosely bound, you can see that the most efficient binding partner would be aspartate. And then the moderate binding would be glutamate, for example. And one bond is better than two hydrogen bonds. So it may be caused by steric interactions. And there's many things that you can see. You can also see the drug design. So for this one, it shows you that there is certain parts that are conserved. And if you measure all the distance inside the pockets, there is certain part that should be expanded or should be contracted in the pocket in order to get better drug, for example. So there's many things you can do with the server. So that's just the demo of how it does. So it can also go to the variant, to the SNPs like this one. So it highlights the location where it was mutated. And then just finish the assignment, you should be okay. And if you have further queries, you can also email me, or you can post at Google Class. For the real bioinformatics, you need to hands-on and you actually put in the data and then get the output and then interpret the data. But this one will be the basics that you understand the whole thing, so you can relate all the information from central dogma to the concept that we want, the similar in sequence and fold and infer the function, and then we can link out to the universe of them, to various pathways, to drug design, to everything. So this one will be the core of everything. I think it should be fine. So let's stop the meeting now, and then thank you for your attention.
Thank you, Ajarn.
ค่ะ โอเคค่ะ สวัสดีค่ะ